Welcome to our journey into the microscopic world of cell biology. Today, we are exploring one of the most fascinating organelles inside eukaryotic cells, the Golgi apparatus. Think of it as the ultimate shipping center of the cell. Understanding how this organelle functions is absolutely crucial for grasping how our bodies maintain order, process vital molecules, and keep us alive every single day. Let us dive in and see how it works. To start, let us build an intuitive understanding of what this organelle actually does. Imagine you just ordered a package online. Before it reaches your doorstep, it has to go through a massive sorting facility where workers check the item, put it in the right box, slap a shipping label on it, and send it out on a delivery truck. In the microscopic world of your cells, the Golgi apparatus acts exactly like this bustling post office. It receives raw, unfinished proteins from the factory, which is the endoplasmic reticulum, modifies them, packages them, and ships them to their final destinations. Without this incredible sorting center, the cell would be in absolute chaos, with proteins wandering aimlessly and never reaching the places where they are needed to keep you alive. Now that we understand its overall purpose as a post office, let us look at its physical structure. Scientifically, the Golgi apparatus is a highly structured organelle found in eukaryotic cells. It consists of a series of flattened, stacked membrane pouches called cisternae. You can think of these cisternae as a stack of hollow pancakes. Each of these individual pouches is not just empty space. They contain specific enzymes that perform distinct chemical reactions. As proteins move through these layers, they are progressively modified, ensuring they are perfectly prepped for their specific roles in the body. Building on that pancake-like structure, it is important to know that this organelle has a strict directional flow. The Golgi has a distinct polarity, meaning it has a receiving side, called the cis face, which points toward the endoplasmic reticulum, and a shipping side called the trans face, which points toward the cell membrane. As proteins and lipids travel from the cis face through the medial or middle region to the trans face, they undergo sequential modifications. A major one is glycosylation, which is the addition or removal of sugar molecules. Finally, these mature molecules are packaged into small membrane-bound bubbles called vesicles, which bud off from the trans face and travel to their specific cellular targets. To make these structural concepts concrete, let us look at this diagram. Here, you can clearly see the stack of flattened membrane pouches, or cisternae, colored in light blue. Notice the distinct polarity we just discussed. At the bottom is the cis face, the receiving side, where small transport vesicles arriving from the endoplasmic reticulum are fusing with the Golgi. As you follow the large central arrow upwards, you move through the medial cisternae, where chemical modifications happen. Finally, at the top, you see the trans face, the shipping side. Here, you can observe the newly formed secretory vesicles budding off, carrying the fully processed proteins away to their final destinations. Having seen how it operates, you might wonder why the Golgi apparatus matters so much in real life. Consider the cells in your pancreas that produce insulin, a hormone essential for regulating your blood sugar levels. The Golgi apparatus in these cells is responsible for processing the raw insulin protein, packaging it perfectly, and ensuring it is secreted into your bloodstream at exactly the right moment. If the Golgi apparatus were to fail, your body could not secrete insulin, digestive enzymes, or antibodies to fight off infections, leading to severe diseases. Furthermore, many genetic disorders, such as certain forms of muscular dystrophy and Alzheimer's disease, are linked to defects in how the Golgi apparatus sorts and modifies proteins. While biology is often descriptive, this incredible efficiency leads us to a mathematical model of how the Golgi ships out proteins. We can define a simple equation to calculate the total secretion rate. The formula is R-sec equals NV times PV, all divided by T. Let us break down these symbols. R-sec represents the secretion rate, measured in proteins per minute. NV stands for the number of vesicles budding off from the trans-Golgi network. PV represents the average number of protein molecules packed inside a single vesicle. Finally, T represents the time interval in minutes. This tells us that the total rate of secretion depends directly on how many delivery trucks, or vesicles, are leaving and how fully packed each truck is. If the cell needs to secrete more hormones, it can either increase NV by making more vesicles or increase PV by packing more proteins into each one. Now that we have our formula, let us apply it to a real biological scenario. A pancreatic beta cell needs to secrete insulin after a meal. We are given that the Golgi apparatus produces 150 vesicles every 5 minutes, and each vesicle contains an average of 2,000 insulin molecules. We need to find the secretion rate per minute. We start with our formula and substitute the given values. The numerator becomes 150 times 2,000, which gives us 300,000 total insulin molecules packaged in that 5-minute window. Dividing this massive workload by 5 minutes gives us our final answer. The Golgi is successfully processing and secreting 60,000 insulin molecules every single minute. This high rate shows just how incredibly efficient and vital the Golgi is for maintaining your blood sugar levels. Having successfully used the standard formula, we sometimes face situations where we know the required secretion rate, but need to find out how hard the Golgi must work to meet it. To do this, we must rearrange our equation to solve for NV, the number of vesicles. We start with our original secretion rate formula. First, we multiply both sides by the time, T, to clear the denominator. This gives us the total number of proteins needed. Next, we divide both sides by PV, the capacity of a single vesicle, to isolate NV. This algebraic rearrangement is a smart strategy because it isolates the exact variable we are trying to find, preventing calculation errors later. We now have a direct formula for the required vesicle production. Let us put our newly derived formula to the test with an immune cell fighting an infection. The cell must secrete 120,000 antibody proteins per minute. The Golgi can pack 400 proteins per vesicle. We need to find the production rate per second. First, we use our rearranged formula and plug in the required rate, the time of 1 minute, and the capacity of 400. Dividing 120,000 by 400 tells us the Golgi must produce exactly 300 vesicles every minute. But the problem asks for the rate per second. So we take one final step and divide those 300 vesicles by 60 seconds. We arrive at 5 vesicles per second. This means the Golgi is rapidly assembling and shipping out 5 separate delivery vesicles every single second to keep you healthy. That staggering calculation we just completed brings us to a profound realization about cellular biology. The fact that a single organelle can produce 5 transport vesicles every second beautifully illustrates the dynamic, non-stop nature of cellular organelles. It proves that the cell is not just a static balloon of fluid, as it is sometimes drawn in simple diagrams. Instead, it is a highly active, bustling factory that never sleeps. The Golgi apparatus sits at the very heart of this continuous activity, constantly adapting its processing speed and vesicle production to meet the immediate physiological demands of your body. We have covered a lot of ground today, so let us summarize the most important ideas. First, the Golgi apparatus is the indispensable shipping and receiving center of the eukaryotic cell. Second, it takes raw proteins from the endoplasmic reticulum, modifies them with chemical tags, and sorts them into vesicles for precise delivery. Third, we learned that this incredible efficiency can be modeled mathematically, showing how vesicle count and capacity determine secretion rates. Finally, whether it is secreting insulin or antibodies, the flawless operation of the Golgi is what keeps the complex society of cells within your body functioning smoothly. You now have a solid mathematical and biological understanding of this vital organelle.